What I'm going to do here in this video is I'm going to start off by talking about the process of contraction in cardiac muscle tissue overall, and then we'll go back and look at each step individually, okay? So this is the overall slide. This is a summary of what happens during cardiac muscle cell contraction. So it all starts with the autorhythmic cardiac muscle cells. Unlike skeletal muscle tissue, cardiac muscle tissue does not require nervous system input to contract. That doesn't mean we don't have nervous system input coming into the heart, but we don't need it, okay? So we have those autorhythmic cardiac muscle cells that are able to self-start action potentials. So they can self-depolarize and then initiate an action potential in themselves. The action potential then can spread to the normal contractile cardiocytes. So don't forget that cardiac muscle tissue has those intercalated discs where one cardiocyte ends and the other cardiocyte begins. There are these intercalated discs between them. And the intercalated discs allow the cardiac muscle cells to share a cytoplasm. So whatever happens to the cytoplasm of one cardiocyte will also happen to the next. So when the autorhythmic cardiocyte initiates an action potential, it can spread to the contractile cardiac muscle cell, and then it can spread to its neighbor, right? What's happening here are ions like calcium and sodium are flowing through tunnels in the intercalated discs and just following their diffusion gradient, going from an area of high concentration to low concentration, traveling from one cardiac muscle cell to the next. The goal of the action potential, which is a traveling change in voltage, by the way, I didn't mention that, right? The goal of this traveling change in voltage, the goal of this action potential, is to bring calcium into the cytoplasm of the cardiac muscle cell. And it's able to do this in two ways. The first way is through use of voltage-gated calcium channels. So the action potential is a traveling change in voltage. That traveling change in voltage will open voltage-gated calcium channels that are embedded in the cell membrane of the cardiac muscle cells. Once the voltage-gated calcium channel opens, calcium then can diffuse from high concentration outside of the cell to low concentration inside the cell. So the calcium just diffuses into the cytoplasm of the cardiac muscle cell. Another way we can get calcium into the cytoplasm of the cardiac muscle cell is to open up our storage sacs of calcium, the sarcoplasmic reticulum. So in order to open the sarcoplasmic reticulum, basically we first have to get calcium into the cardiac muscle cell. I know it sounds ridiculous, but turns out there are these proteins that are embedded in the membrane of the sarcoplasmic reticulum. And these proteins are calcium sensitive. So once the calcium enters into the cardiac muscle cell through the voltage-gated calcium channels, that calcium then triggers these proteins to open, and the calcium will diffuse out of the sarcoplasmic reticulum. This process is called calcium-induced calcium release, which sounds ridiculous, but it works. Okay, so we just work really hard to get calcium in the cytoplasm. So why do we need the calcium in the cytoplasm? If you said because calcium binds troponin, you're right. So calcium binds to troponin, which will shift when the calcium binds. And that will shift the attached tropomyosin, revealing the binding sites on actin. Once the binding sites on actin are revealed, myosin can grab onto the actin and pull, producing the actual movement associated with contraction. All right, and that is the whole process in a nutshell. So now let's take a look at some of these bits and pieces in more detail. We're not going to go into massive detail on the autorhythmic cardiac muscle cells because they're super complicated and we don't really need to know all those details anyway. But what we should understand is the fact that the autorhythmic cardiac muscle cells are capable of self-depolarization. In order to get an action potential, you need a depolarization. So the self-depolarization is what is going to lead to the action potential, right? So action potentials are traveling changes in voltage, and they are started by a depolarization, okay? And that's followed by a repolarization. So the repolarization is basically the voltage kind of going back towards normal. Right? So the action potential that occurs here in the cardiac muscle cell is slightly different than what we saw with neurons and skeletal muscle cells. But we still have a depolarization and a repolarization. And if you need a recap on those terms, they're over on the right-hand side of the screen. Right? So the depolarization and the repolarization process involves a lot of moving ions. And because ions are charges, these are moving electrical currents, right? And we can actually see this moving electricity on an EKG. And we'll talk more about EKGs later. At the bottom of the screen, we can see a graphical representation of an action potential for an autorhythmic cardiocyte. And we don't need to know this graph for testing purposes, but it does give us some information, and sometimes looking at a graph can be really helpful for understanding information. So on this graph, we see basically three types of ions that are important for this action potential: sodium ions, calcium ions, potassium ions. Don't forget that sodium and calcium are highly concentrated extracellularly, whereas the potassium is highly concentrated intracellularly. So if you open up a sodium or a calcium channel, the sodium or calcium will go from outside the cardiac muscle cell to inside. But if you open up a potassium channel, the potassium will move from inside to outside. Remember, the ions just follow their diffusion gradient, their concentration gradient. The autorhythmic action potential begins with a depolarization, which is mostly due to the opening of voltage-gated sodium channels and sodium rushing into the cardiocyte. This is then followed by the opening of voltage-gated calcium channels and calcium flowing into the cardiocyte. Then we go into our repolarization phase, where we open our voltage-gated potassium channels, and the leaving of the positive potassium is what causes the repolarization. Here we can see a visual representation of how ions can diffuse from one cardiac muscle cell to the next by going through those intercalated discs. Now, this image is highly simplified, but it gets the point across, okay? So in the picture here, we see two cardiac muscle cells with an intercalated disc between. The little red circles are sodium ions. The red wedge is showing you the sodium gradient, so there's more sodium outside than inside. And then we also have a voltage-gated sodium channel in the cell membrane. So during the action potential, the voltage-gated sodium channel will open, and then the sodium ions will diffuse from the area of high concentration towards the area of low concentration inside the cardiocyte. Now, don't forget, this cardiocyte is attached to the next through the intercalated disc. So this is a low sodium area. So the sodium will diffuse from the one cardiocyte to the next by going through the intercalated discs. And we can very quickly move ions in this way. Every cardiac muscle cell is connected to its neighbor through an intercalated disc. It's kind of like every cardiac muscle cell is connected to every other cardiac muscle cell. And so they're kind of like just one big giant cell. And you would think that the entire myocardium would contract at once because of that. But it doesn't. The upper cardiac muscle cells contract together, and the lower cardiac muscle cells contract together. And this is because of the fibrous skeleton of the heart called the cardiac skeleton. So the cardiac skeleton prevents the ions from flowing, the electricity from flowing from the atria down to the ventricles. So the upper portion, the atria, contracts together, and then the lower portion, the ventricles, contract together. This requires little patches of autorhythmic cells in two different places in the heart. We're going to need autorhythmic cells to start the action potential for the upper chambers, and then we need another patch of autorhythmic cells to start the action potential for the lower chambers. And we talk about this in a lot more detail in another video when we look at what's known as the cardiac conduction system. So these little groups of autorhythmic cells are called the SA node and the AV node. Here we can see the process of getting the calcium into the cytoplasm. And that's the whole point of the action potential, is to get calcium into the cytoplasm. Let's take a look at the image on the screen. This is our cardiac muscle cell membrane. So this would be outside the cell. This is inside the cell. Here is the membrane that makes up the sarcoplasmic reticulum. Right? So this is inside the cell. This is the inside of the sarcoplasmic reticulum where we store that calcium. And the two important players here are this voltage-gated calcium channel and then this calcium-sensitive channel. We're going to open both of these to get the calcium into the cytoplasm. So the action potential is that traveling change in voltage that travels along the cell membrane. And when the action potential hits this voltage-gated calcium channel, it will open and calcium will rush into the cardiac muscle cell. That calcium will then bind to this channel, opening it, allowing the calcium to diffuse out of the SR and into the cytoplasm. So that was the calcium-induced calcium release that I had mentioned previously. The voltage-gated ion channels account for about 20% of the calcium that gets into the cardiac muscle cell, but the majority of the calcium will come through the SR via that calcium-induced calcium release. It's about 80%. Some people will use a calcium channel blocker as a medication. So what kind of effect do you think that would have on the heart if we used a calcium channel blocker? So we would block this thing right here, right? So we wouldn't get that calcium coming in. And basically, this is going to stop the whole action potential process. And it really does help to decrease the contractility and the contraction strength of the heart. So we got the calcium in the cytoplasm. Now what? What was the whole point of that? So this slide is describing what we're doing with the calcium. The calcium is needed to bind to troponin to move the tropomyosin off the binding sites on actin, and this will allow the myosin to grab onto actin and pull. And that's what we can see in the pictures here. This is what it looks like in the relaxed state, and this is what it looks like in the contracted state. Notice that the calcium is bound to troponin, and that's moved the troponin, which has moved the tropomyosin off the binding sites on actin, and now myosin can grab onto the actin and pull. We've looked at the importance of the action potential. We've looked at the importance of calcium. Now we have to talk about the importance of ATP for proper myocardial function. The process of myosin grabbing on to actin and pulling involves ATP. We're not going to go into the nitty-gritty details here, but let's just say that in order for the myosin to grab on to actin and pull and then release and then grab onto another actin and pull and release, something in that whole process requires ATP. ATP is mostly made through a process called aerobic respiration, also known as cellular respiration, and aerobic means in air. So this means it requires oxygen. So aerobic respiration requires oxygen in order for us to make ATP. It also produces a waste byproduct gas called carbon dioxide. So we take in the oxygen to make the ATP, but once the ATP has been made, we have that waste carbon dioxide that we have to get rid of. What would happen if cardiac muscle cells experienced a lack of oxygen? If they didn't have enough oxygen, they wouldn't be able to do the aerobic respiration, which means they wouldn't be able to make ATP, which means they wouldn't be able to have the myosin grab onto actin and pull and then repeat, which means they wouldn't be able to contract. So ATP is absolutely essential for cardiac muscle cell contraction.